Okay, guys, hi, I'm Ruggeri. I'm a PhD student at USC, and today I'm going to be talking to you guys about the ongoing work I'm doing this summer, which is on thermal tolerance in symbiotic anemones across an environmental gradient, and we'll get more into what that means. But first, I want to just talk to you guys about my journey of how I actually got here. So I grew up in New Jersey, pretty close to the coast. This is me right here. So my family were pretty much beach bums, so we spent like every day in the summer at the beach. All of our vacations were pretty much centered around the beach, the ocean. So I went to a lot of places in the Caribbean, got very comfortable in the water, snorkeling around, interested in marine life early on, but I had no idea that this could ever turn into a career. So actually, when I was going off to college, I was choosing between going to art school in New York to do photography or psychology at the University of Miami. And after visiting the University of Miami, it kind of became like, there's no choice here. It was so beautiful. I was so drawn to it. And I didn't know at the time, but looking back, I really chose the school because of the proximity to the ocean and that I could go in the water year-round and go to the reefs and snorkel around. But I was a psychology major still, so spent about a year studying that, but still got drawn to the marine side. So I thought about switching majors, but I was kind of like, I don't know, I don't want to make a huge decision. So I volunteered at a marine science camp in New Jersey over the summer. So that's not me up there, but it's someone else doing fish dissection. So we taught kids about marine science, and they really asked just the most interesting questions. So it really inspired me to just be more curious and really set in that I want to learn more about this system, more about marine science. So I switched my major to marine science and biology, and I started working in a lab. This is me right here. So doing fish surveys. I was doing fish surveys to determine the habitat value of human-made structures, so things like docks and seawalls, compared to natural mangrove habitats. And then also, the other side of my lab worked with coral restoration. So I also got experience going out helping them outplant corals, which kind of made me more interested in symbiosis and to study this anemone right here. And that will all make more sense to you as I tell you a little bit more about this system. So I study the aggregating anemone, Anthopleura elegantissima. It lives in the intertidal zone, so a very extreme environment. When it's exposed, it has to deal with thermal stress, light stress, wind stress, you name it. So they're very hardy. They can reproduce both sexually and asexually. So up here I have, this is one reproducing asexually. So it pretty much just walks away from each other and splits into two, and they're genetically identical. And so what they do is they just keep doing this until they form these large aggregations. So this is 100, maybe more anemones in this aggregation, and they're all genetically identical. Like I said, they also reproduce sexually, so you can find different aggregations that are genetically distinct from one another. But within one, they're all the same. They're all clones. Another thing that's really interesting is that they have symbiotic algae that lives within their tissues. So symbiosis, in this case, is a mutually beneficial relationship between the animal, the anemone, and microscopic algae. So that's a picture under the microscope that I took of just a tentacle grounded up, and these are the algae that live within it. So like plants, these algae use sunlight and CO2 and convert it to photosynthetic products like sugars that the anemone can use as food. And then the anemone in turn provides shelter for the algae. So it's free from predation. It has a nice environment that's exposed to light so that it can do its thing. However, when they get stressed out, they actually lose their algae. So you may have heard of coral bleaching. So corals are the foundation for coral reefs worldwide. And they also have symbiotic algae that live within them. When they get stressed out, the algae either leave or they reduce their photosynthetic pigments, which results in a bleached animal. So it looks white, which is why they call it bleaching. So this could be due to a number of environmental stressors. Most notably, I think, is rising sea surface temperatures, which is due to global warming. And this is causing declines in coral reefs worldwide today. So what I'm interested is looking at the genetic and the environmental factors that lead to increased thermal tolerance in these symbiotic anemones. So I'm making two main comparisons. One is between the high and low intertidal. So you can see the intertidal forms this vertical gradient here, whereas if you're higher up, you're getting exposed more when there's low tides and being exposed to higher temperature, so air temperatures here, which can reach, we all know, like 190 degrees very frequently, whereas when they're submerged, it's very cold, like 60 degrees. So huge thermal fluctuations that they're experiencing, whereas the ones that are mostly submerged are only in a more stable environment, only being exposed to a colder temperature. So I'm trying to see if there might be selection for more thermally tolerant genotypes in the higher intertidal. And it also could be not selection, so it could be selection for the actual genes that are conferring thermal tolerance. Or it could just be that the ones in the higher intertidal are used to it. They're getting exposed to high temperatures a lot, so they can increase their thermal tolerance. And then the other comparison that I'm making is within genetically identical aggregations. So right here, all the same clone, someone actually found that if you measure the body temperature of the ones on the outside during tidal exposure, they are heating up more than ones on the inside, which are kind of buffered from the environment. So non-genetic mechanisms, so just from conditioning to high temperature versus conditioning to a lower temperature, are there differences in thermal tolerance within these aggregations? So now I'll just show you, this is some of my preliminary data. So not a lot of people work with these anemones, actually. I don't know of anyone in Southern California who has. So my first thing I had to kind of figure out, which is what temperature are they going to start bleaching? Are they going to start expelling this algae? So I exposed them to 5 different temperatures, 18 being the control temperature. What they'd be, it's 60 degrees Fahrenheit-ish. So what they'd just be if they were submerged, versus higher temperatures, 25 to 31, and every 2 degrees in between. And then I measured chlorophyll, which is a photosynthetic pigment. So that will tell you either the amount of symbionts kind of in the tissue, or if they're just reducing those pigments during stress. So as you can see, it worked. As you increase the temperature, they're decreasing those photosynthetic pigments, so they are bleaching. And you can actually see this visually, which is kind of cool. So up here you have one that has its symbiont still, and this one that has bleached. And what I was really looking for is kind of the temperature that I can use for my full-scale experiment, where you're going to see significant differences. And so that's going to be around 29, 28 degrees. Then I went on, so this is just preliminary data, a pilot study, but I separated the high individuals, the ones collected from the higher intertidal environment, from those collected from the lower intertidal environment. And as you may recall, the idea is that ones in the higher intertidal would be more thermally tolerant than those in the low intertidal. This is showing exactly the opposite. Exactly the opposite. So the ones in the low intertidal, the blue, they bleached less than those that were sampled from the high intertidal. Now, like I said, this is a pilot experiment, so not that many data points. I should have more to really tease this apart, and I'm not sure why this is yet, but one thing I could say that it could be is just the ones in the high intertidal, they're exposed to so much stress, other kind of stressors that they just can't handle another increase in temperature. They just expel their algae. But I'm not sure. It's something that I need to look into more and should have more answers when I run the full experiment. But really the interesting thing here is I need to find a temperature where I start to see this variation. So if I chose too high a temperature, they might all just bleach, they might all die. And too low, you might not see this variation. So by doing this, I kind of got to choose a temperature where I start to see them separating here. So the next step, so tomorrow and Monday, actually I'm going to go out into the intertidal. I'll be collecting 240 anemones from the high and low, as well as within the aggregation and the outside of the aggregation. And so I'm going to be exposing them to 28 degrees Celsius, around 84, I think, degrees Fahrenheit, where we saw the bleaching happen. And then I'll be phenotyping these, which basically just means measuring certain traits, mostly that have to do with bleaching. So the number of symbionts that are in the anemone, the photosynthetic pigments. I'll also be doing photosynthesis measurements itself. And then the last thing I'll be doing is genotyping the anemone. So sequencing the host animal as well as its symbionts to see if that is a determining factor of their thermal tolerance. So the host can be more thermal tolerant and keep their algae, but also the symbiont itself, if it's more thermal tolerant, it might be able to stay in the anemone despite the host being more thermally tolerant. So this is something I'm going to try to tease apart from this data. So with that, I'd like to acknowledge my advisor, Carly Kenkel, the Wrigley staff, especially Kelly, who helped me set up all of my experiments. And then I was also funded by the Wrigley Bakus Fellowship, so I'd like to thank them. And I'll take any questions. Yeah?
What's the process to get out there and collect them?
The process, so I go during the low tides, basically. So tomorrow it's like negative 0.5 feet, which means it's below the mean low tide. And that way it's easier because you could just walk out, they're exposed, and you just gently pry them off the rocks. And I'll just put them in baggies that have water and take them back to the lab here and put them right into aquarium. Yeah.
Besides the bleaching, what are you seeing like with reproduction or disease tolerance, that kind of stuff?
So that's very interesting. We don't know yet for these anemones. I'm not sure. So one thing I'd like to get them to try to asexually reproduce in the lab, which would be amazing, but it seems to other people I've talked to, it takes a long time to do that. But generally for corals, if they are bleached, they're more susceptible to things like disease, and I mean, they usually die, so reproduction's kind of off the table at that point, but it's very difficult in corals and even the anemones to study reproduction, or at least sexual reproduction, because they're really long-lived. So it takes a long time to try to do experimental crosses and then grow them up to another reproduction.
Can't tag them or anything.
You can tag them, but the thing is, and you can get the larvae, but if you want to make them grow again, it takes a long time for them to get to a size big enough for them to reproduce again. Yeah.
So under stress, the anemone expels the algae or the algae leaves? And if it leaves, where does it go? Or does it die and just...
That's a great question. So one thing is that I can do is look at, so like I said, I just do cell count. So like you saw, like under the microscope, count the number of algal cells left. So there's a couple kinds of bleaching. It all leads to them looking white, but it could be from, right, the host expelling the algae, could be from the host degrading the algae, so like essentially eating it, breaking it down, eating it, could also be from the algae itself, just reducing its photosynthetic pigments because they're stressed. So there's a couple ways, and we can kind of get at that by looking at the number of cells versus the chlorophyll pigments to see if it's like the algae or they're actually just not in the host.
So at the end, you were saying something that you were going to study whether whether there's the anemone or the algae that was stressed to start this, or is it both of them? Is that something you're looking to do?
Yeah, so we can try to make correlations between the genetics of the host animal and the thermal tolerance that we see, as well as the symbiont genetics, and if that's driving like what we're seeing between different aggregations, or within an aggregation would be great if there are different symbionts. Then you can really tease apart if it is that symbiont that's driving it. But yeah, it's difficult to. Genetics is really the only way I can think to kind of get at that question more. Some people have cultured the algae and so do the same thing where they expose the algae itself to higher temperatures and look how they react versus inside the host, because they can be free living. You can culture the algae outside of the host, but it's a different environment and we can't really mimic the environment that it would be within the anemone, so it's very hard to get at those questions, yeah.
I just have a couple questions.
Sure.
One, is the algae species specific? Is it the same kind of algae in these as you would see in any coral?
So it's not the same species itself, but they're within the same family.
Are they interchangeable, I guess I'm wondering? Are they interchangeable? Or
So I'm not sure as far as like these specific species to coral. I don't know that specifically because I mean these are found on the west coast.
Right.
I'm not sure.
Yeah.
I would have to
And
look
then
that
the
up.
other question I had, I don't know, I thought I heard this somewhere, but like with global warming and warming sea temperatures, doesn't that affect the acidity? And I'm wondering if like the acidity is a factor in like what happens with these pigments.
So for corals, that's a very big question because they have skeletons, and so their skeletons will actually start to dissolve in acidified conditions. There was one study that exposed these anemones to acidified conditions, and they didn't really care. So they were fine with it. The thing is that it could be is sometimes these anemones, when they're in like tide pools, so, you know, the water washes away and they're just in this stagnant water, that they are just exposed to more acidified conditions from all of the animals in the tide pool respiring CO2.
Right.
So they might just be adapted to it. But yeah, for corals, that's a very big concern.
I was wondering, you know, they do this kind of reforesting with corals, but then what keeps them from bleaching?
Right.
I don't get
there
that
isn't.
part.
So I mean, there's nothing that keeps them from bleaching. One thing that people can do is kind of experimentally determine which corals might do better in that sort of condition, and try to outplant.
If you could find like those algae that could survive those kind of temperatures, and then if those algae could be inoculated into different corals, and maybe
I
that
mean,
could
you might as well just start studying this. Those are questions, that are really
Interesting.
big. There definitely are more heat tolerant algae. Sometimes they're not as productive as other algae, so they're not producing as much food for the host. The other thing is a lot of corals, these anemones can be specific to their partner, so they'll preferentially have one symbiont over another, regardless of heat tolerance. We don't necessarily know
It's going to
why.
be great CO2 sponges, right? All that algae.
Yeah.
Yeah.
Sure.
How did the host and the symbiont interact? I mean, the symbiont is benefiting the host, and probably vice versa. Probably just the structure of the host helps the symbiont to be in a place where it can food is coming by. But how does the host get something from the symbiont? Is there a membrane or what?
So the algae are actually inside of this, they call it the symbiosome. So it's like a specialized cell that's a host cell. And so they actually they can be free living, like I mentioned, in culture, but when they're inside the anemone, they lose that ability. So they're just within these specialized cells, and they will transport sugars to the host through that membrane.
Oh, still like an organelle.
Yeah,
Yeah.
almost. Yeah. In the, okay,
okay.
whichever.
Is it sunscreen? You know, I hear about sunscreen in Hawaii. Is it doing the same effects? Like,
Oh, that sort of sunscreen. I don't know.
you know,
So the only thing I would say is, sunscreen obviously blocks sunlight. So if these need sunlight, which they do for the algae to really photosynthesize, then it's going to, have an effect. As far as the research on sunscreen, I can't speak to it all that much. It would definitely have an effect, but I don't think there's been enough of people going out into the environment and measuring concentrations of sunscreen actually in the water.
but I think in Hawaii they banned.
They
that.
banned it,
Yeah.
yeah. It's actually a huge debate. So it's a huge debate, yeah. Sure.
I've noticed when walking along tide pools that the aggregates, the colonies are separated usually by a certain
Yeah.
way. Are you taking from different colonies or just one?
Yes, so different colonies to get ones that are genetically different as well as within the same colony. So I can expose like the same clone to different conditions to reduce the variation. But yeah, it's really interesting. So the clones, if they're genetically different, they'll actually fight each other and one will kill the other. Yeah, so it's very weird. Exactly. And so when I have them in the aquarium and stuff, I need to put them in like these little cups as a barrier so that they don't kill each other and I just end up with one clone. Yeah.
You might have said it before we got here. I apologize if I asked you to repeat yourself. What was the sample size of your pilot experiment?
So this pilot study, so that was 8 anemones per temperature treatment. So not that much.
Right.
This is going to be 240. So really scaling up just to really get at that temperature because I wouldn't want to collect 240, expose them to the wrong temperature, and then not have
Mitigate
enemies.
those.
Yeah.
Have you tried to bring the anemones back after the bleaching by returning them to the cool temperature?
I have not. That is really interesting question. I'm not sure. I'm sure they would recover. So these anemones, unlike corals, actually, they can survive without their symbionts. So without the algae, they can survive. Obviously, when you heat them up, it's a stressful condition, so it's an energetic expense. But depending on the temperature, if it was a really mild stress, I would assume they would be able to recover. And, corals can recover from bleaching sometimes too, if that's the case.
Amazing. Oh, one more.
Oh, I just The
Sure, yeah.
The word. It's the
word.
the
So-
C word.
Sea lab, that's
Oh, right
it's
below C. That's the
So
Nadarian.
It's
How do you-
Cnidarian. So Cnidarian is basically the phylum that anemones and
Okay, so
corals
it's-
and
okay,
jellyfish
right,
are in. So we're the Cnidarian Evolutionary Ecology Lab.
That's a mouthful.
Well, sea lab.
Yeah.
Not that big. And then the others tiny, tiny.
I like the
That's a
plan.
spelling bee.
CNC.